The WIN consortium first came together really as a vision of Vladimir Lazar, the chief operating officer, along with the support of Thomas Turs, who was at that time the Director General of Institut Gustave Roussy, and John Mendelsohn, who was at that time the president of the MD Anderson Cancer Center. And this was in about 2008 when the vision of having a global precision oncology research network was formed. The consortium itself was not formally incorporated as a not-for-profit entity in France until a few years later. So this year's the 10th anniversary of the WIN Symposium and about the 8th anniversary of the formal formation of the WIN Consortium. So I think it's worthwhile to just take a minute to distinguish between the WIN Consortium and the WIN Symposium. The WIN Symposium is an annual scientific symposium. And it has, I think, grown over each of its 10 years, both in terms of the quality of the presentations at the symposium, the attraction to the symposium of really major global figures in oncology and precision medicine in particular, the quality of the scientific abstracts presented at the symposium by the meeting participants, and the overall size and scope of the symposium has continued to expand and become much higher quality scientific meeting. The consortium itself has also grown substantially over this span of time with the addition of new members, both academic centers, clinical centers, pharmaceutical company partners, technology company partners, and so on. Most importantly, I think, and to be featured at this year's symposium, is really the development of now a number of very innovative precision oncology clinical trials that are being deployed now across all of the WIN Consortium clinical centers to hopefully demonstrate the impact of bringing precision oncology directly to the forefront of cancer care. So the overarching theme is really the global implementation of precision oncology, and with the aspirational goal of winning the war against cancer. But I think each year the symposium has focused more and more toward the actual implementation of precision oncology, both in clinical trials and at the level of practicing oncologists. And this year, I think a lot of the symposium is really built around the launch of this series of WIN-sponsored clinical trials that are being launched at WIN member institutions all around the world. One of the advantages of that, of course, is the opportunity for WIN, unlike most any other translational research network, to have access to and to study the diversity of the human population in different parts of the world, in different countries around the world, with different genetic makeups, different cultural habits, and so on, so that we can fully both embrace that diversity and understand its impact on the optimal treatment of cancer and on the effectiveness of various precision medicine approaches. So I think what we'll see at the symposium this year is discussion about the one WIN trial. The WINTHER trial has been completed now and will be reported in advance of the WIN symposium, so it'll be able to be talked about in some depth at the WIN symposium. And then the series of WIN trials that are about to launch or recently have launched. The SPRING trial has already launched. The booster trial has launched. The WIN taper trial is aiming to be launched by the end of this year. Each of these addresses a novel facet of precision oncology, and we'll overall hope to achieve the vision of the symposium this year of global implementation of precision oncology. To a great extent, I think the rapid progress that we've seen in cancer in the last 10 or 15 years has really flowed from a much more in-depth understanding of the biology of cancer, particularly its genomics, and particularly the interplay between the host, particularly the immune system of the host, and the tumor, the tumor ecosystem itself. And as it's become more and more well known that cancer is largely driven by genetic mutation, and as we've come to understand and been able to assess and measure those mutations and understand the mutational burden in cancer and how specific mutations drive a cancer, and correspondingly then have been able to develop effective drugs that interrupt those genomically driven pathways. We've seen the introduction of many, many highly effective, highly targeted anti-cancer drugs that are producing far better outcomes for patients than at any time in the history of cancer treatment, with better results, less toxicity. And the key to a lot of that is understanding which drug to use in which patient by the assessment of the genomic alterations in the tumor with well-validated biomarker tests. We've now begun to see the convergence, I think, of genomics and immunology in that a lot of the immuno-oncology treatments turn out to be particularly effective in tumors that have lots of mutations. And in part, that's thought to be because all those mutations result in the production of lots of novel and mutated proteins on the surface of tumor cells that then can be recognized by the immune system as the immune system is turned on or enabled to find and attack the tumor. So it's a very interesting time in cancer right now, largely driven by a much deeper understanding of tumor biology. That said, that is also still the greatest challenge in really bringing precision oncology to routine clinical care, because number one, we still have a lot to learn. As we do more and more genomic profiling of tumors, we find lots of mutations, the significance of which is not well understood. We don't know if these mutations are really drivers of the tumor or just coming along for the ride. We don't know whether they are targetable with available therapies or therapies that are in development. And doctors, of course, are having a lot of trouble understanding how to interpret these genomic test results and trying to use that to direct patients to particular therapies. So if the field is going to move forward, we need to both continue to do basic research and understand the biology and the clinical implications of the genomic alterations in the tumors, and then to provide a lot of education and clinical decision support to physicians so that they can better understand these test results and use them in the best possible way to guide patients toward what we hope to be the most effective therapies for each individual person. So the SPRING trial, in a sense, is really a biomarker validation study. The SPRING trial builds on the concepts of combination chemotherapy that really extend back to the early years of successful therapy for tuberculosis, where it became recognized early on that a single antibiotic was not going to be effective in curing most patients with tuberculosis, but that by combining several different antibiotics with different mechanisms of action, you could get cure of that disease. The same concept was brought forward in the early days of combination chemotherapy for cancer, where we quickly learned that for leukemia and Hodgkin's lymphoma and other kinds of lymphoma, that combining several effective anti-cancer drugs all at once could lead to a curative therapy for those types of cancer. The same concept has proven effective in HIV/AIDS, where it's become clear that combining multiple anti-HIV therapies in in a multi-drug course of treatment can lead to prolonged control of HIV infection and essentially elimination of the clinical manifestations of AIDS. So now that we have multiple targeted therapies and we're beginning to understand how these different biological pathways interact in driving a cancer, the next logical step forward is to say, we're not going to get long-term control of cancer with inhibiting one pathway at a time. We have to inhibit multiple pathways simultaneously. The question then becomes, what are the pathways that need to be inhibited in any one person's tumor to get the optimal effect? Because it's unlikely that all the same pathways will be active in each cancer in each person all the time. And if you have lung cancer and I have lung cancer, the 3 activated pathways in your tumor might be different from the 3 in my tumor. So we ultimately need to get to a point where we have tools to be able to determine what is the optimal combination of drugs to use in each individual person. The WIN consortium, largely through the work of Dr. Lazar and others, has developed a predictive algorithm known as the SIMS algorithm to try to determine which drug or combination of anti-cancer drugs is most likely to be beneficial in each person's tumor based upon the way in which the algorithm evaluates which molecular pathways are likely to be overactive in that tumor. So in order to prove that using this algorithm actually produces better outcomes in patients, one has to test this in clinical studies, and SPRING is one of those studies in patients with metastatic non-small cell lung cancer who will have a tumor biopsy performed as well as a biopsy of normal adjacent lung tissue. Those biopsies will be interrogated with a variety of molecular mechanisms. The results of those studies will be put through the SIMS algorithm, and the algorithm will sort of behind the scenes make a prediction as to whether a particular combination of drugs is likely to be beneficial for that patient or not. That information will not be revealed during the course of treatment of patients on the SPRING trial. So while all that's happening in the background, the patients will themselves be treated with a combination of 3 targeted anti-cancer drugs, one that targets the immune system, one that targets vascular endothelial growth factor pathways, one that targets other molecular pathways in the cell that are important for driving cell division forward. And then we'll see when the trial completes what the response rate and what the progression-free survival is of the patients who've gotten those drugs in combination, and will then reveal what the SIMS algorithm predicted about whether or not they would have responded to those drugs. And if the predictive value of the SIMS algorithm is high, if it predicts accurately in a high proportion of patients, we'll then be well on our way to having validated that algorithm as something that can be used then prospectively to assign patients to a particular combination of treatments. So that's, in a sense, the overarching goal of SPRING, is to improve the care of patients with metastatic non-small cell lung cancer by treating them simultaneously with 3 targeted therapies, but in doing so to also validate this algorithmic approach to predicting which 3-drug combination is likely to be best for each individual patient. Well, I think the future for the symposium is very bright. It continues to be really a remarkable collection of sort of luminaries in oncology who come and give state-of-the-art talks to a very receptive audience. Great scientific presentations by young leaders in the field who are bringing their very innovative science to the symposium. One of the unique things about the WIN Symposium, unlike many other scientific meetings, is the fact that the symposium invites presentation from sort of all sectors in the research community. So there are invited invitations from speakers in academia, in the pharmaceutical industry, in the technology industry, from patient advocacy groups. We want to hear all perspectives because, we work in an ecosystem that requires the input of the basic scientists, the clinicians, the commercial entities, and of course the patients to be able to ultimately, develop the best therapies for patients going forward.